Welcome to the Jack Westin MCAT podcast with your host, Phil Hawkins.
And Azaii Muniz.
Welcome back. We're going to be talking about some weird stuff today. And by weird stuff, I mean what's not happening inside you. I feel like a lot of times when we talk about what the AMC wants to test, they tend to focus on like things that are biologically relevant to like humans. And like that's why the passages tend to be about cancer and things like that. And we've been talking about transcription and translation, and those are like really high yield topics on the MCAT. But sometimes the MCAT will test what happens in bacteria. And a lot of their stuff is very similar to what happens in eukaryotes with transcription and translation. But this is something that gets tested a fair amount. And I think it's kind of tricky, not because like the content itself is tricky or like what happens in eukaryotes is tricky. What's tricky is telling them apart and being super careful. Because all the time the AMC will throw a question about some bacteria doing transcription or translation. And the answers, like the 3 wrong answers, are about what's happening in eukaryotes. And so where students will kind of read this like, yeah, that's a thing. But like, you got to be so careful about just pulling apart prokaryote versus eukaryote. So since we've been talking about eukaryotes for the last couple of podcast episodes, figure we should talk about prokaryotes and like what you need to recognize as the difference here. Because the MCAT loves testing prokaryotes.
Yeah, it's one of those things where it's easy to forget that we need to study this. And I don't know about you, but a lot of times when I'm doing comparisons, I find it really helpful to visualize them. I think in general, I find things helpful in terms of learning to visualize them. And this is one of those situations where sitting down, and you don't have to be an amazing artist to do this, but just drawing the differences can be incredibly helpful. Thinking about where things are located, thinking about what types of organelles are not in a particular cell type, and just the key players in each of the processes that we're going to be talking about, I think can be incredibly helpful, especially for some of the later terms that we're going to chat about. They're often mixed up. And so I think it's just super helpful to think about doing that if you have the time.
Yeah. So throughout this podcast episode, we're going to focus on the differences. If something is pretty much the same, we're just not going to talk about it. And so note that there are a lot of overlap between these. Like obviously both prokaryotes and eukaryotes have DNA. They have A's, T's, C's, and G's. Like proteins that they're making are made of amino acids. Those proteins are made by ribosomes. But there are some specific differences. And so we're just going to kind of go through here and highlight the differences because this is what the AMC usually tests. So first off, and this should be the most, one of the clearest things, one of the most obvious differences is prokaryotes don't have membrane-bound organelles. They don't have a nucleus. They don't have a mitochondria. They don't have a rough endoplasmic reticulum. So all of this stuff that happens in eukaryotes is often really compartmentalized, right? Like transcription is occurring in the nucleus. Translation is occurring at the ribosomes, which are usually in the rough ER or maybe the cytoplasm. But in prokaryotes, it's all happening all in the same place. And actually, I want to kind of take a moment to highlight this. On exams, what the AMC will often do is they will mention somewhere in the passage that this organism, this single cell organism, there's this protein that is being translated, and this protein is important for the nucleus. And like, because they say nucleus in the passage, you immediately need to switch your brain to eukaryote. And so all of the things, even if it's a single cell organism, like for example, yeast is one cell, but it is also a eukaryote. Or Plasmodium falciparum, which is what causes malaria. That is a parasite that hides inside your red blood cells, right? So it's inside your cells. So it's really small, but it is still a eukaryote. It is not a prokaryote. And so you got to be really careful with that because I know a lot of times the MCAT will talk about like, something with a nucleus and you immediately need to be thinking eukaryote. Versus if they say something like you have a gram-positive bacillus, you need to be thinking prokaryote. And like telling the difference between those, sometimes that's hidden in the passage itself. And you got to be kind of careful with that.
Yeah, you mentioned something else that is fair game, and that's gram staining. So if what Phil just said, you're kind of scratching your head or not quite as familiar, I would recommend taking a look at that as well. I know we always like to talk about different connection points because you can have a passage that asks about, that discusses the difference between prokaryotes and eukaryotes, but like you pointed out, they can branch into something else. Mm-hmm. So thinking about some of the differences as it relates to the different steps of protein production. So if we remember and we think back to the first episode on this and eukaryotes, we have transcription as the first step. So I think you had that library example and thinking about the instructions. And so this is going to be actually understanding the instructions for building the chair, I believe it was. And so when we're building that chair, we have to think about, okay, well, it's not going to happen in the nucleus. There is no nucleus, right? We have these plasmids that are hanging out in the cytoplasm, and you're still going to have to open up and hold open the plasmid itself. And so one of the differences between eukaryotes and prokaryotes is that we generally think about eukaryotes under the umbrella, or rather having this umbrella of topoisomerases, but there's a particular version that becomes really important with bacteria because our antibiotics can specifically target this, and that's bacterial gyrase, which is within this class of topoisomerases. But I know we've mentioned this before, for things like cancer, for things like bacterial infections, you want to target something that is in that cell type that is either not as prominent in the case of cancer cells versus our own mammal cells, so to speak, or something that is simply not present in our cells. And so this is going to be a way where we can target our antibiotics and have something that doesn't affect our cells, but is really great against infection. And once you're actually in the process of transcription, we'll remember that during that episode, we chatted about how often we say post-transcriptional modifications to remember and to identify some of the stuff that happens after the initial part of transcription. So things like the 5' cap, the poly A tail, but that's not going to happen in prokaryotes. Why? Because they don't have to get out of the nucleus, right? So they don't need that. Protection is already where it needs to be. And in conjunction with that, they don't have any splicing that's going to happen. And so if you think about that process of transcription, remember, those post-transcriptional modifications are still part of transcription. They're just at the very end. So for purposes of the ANC, you've got to know that it's not fully complete until those things happen in eukaryotes, but you don't got to worry about them in prokaryotes, which is nice. It's one less thing to remember for prokaryotes.
Yeah, I do want to kind of highlight this, like the differences between these, and like, as you mentioned, Azaii, like gyrase. Like we in eukaryotes have topoisomerase, they use gyrase as prokaryotes. I always feel like there's always some weird prokaryote exception because there's so many strange bacteria, but we're talking about just generally what's going on here. Anytime there's a difference, that's a target, right? Like this is why this stuff is important, is because all of these things that are different, like their ribosomes are slightly different, their like process of transcription is slightly different, the process of translation is slightly different. And so if we can target those differences, then we can kill bacteria and not kill humans, right? Because if we do something that like just messes with glycolysis, that's going to kill bacteria, but it's also going to kill people. That's called poison. And it turns out that is not a good way to treat a person, is to like, I killed all the bacteria, also the person, but killed the bacteria. Now, we want to kill bacteria specifically in cases where people have infections. So all of that stuff, like no splicing, no 5' cap, no poly A tail, like all of those are important things for you to kind of recognize here. Bacteria also do something like they're polycistronic, or they experience polycistrony, which is a very weird term. If you can spell that in Scrabble, you just win automatically. But eukaryotes, we are monocistronic. So what do we mean by that? So in a eukaryote, like I will have a gene. Now this gene I can turn into a messenger RNA. If I do alternative splicing, maybe I can turn that into a couple different types of messenger RNA. But once I have one messenger RNA, I can turn that into one type of protein. So if I have a messenger RNA that codes for insulin, for example, I send that to a ribosome, I make insulin. And that's it. I can make a lot of insulin off of that one messenger RNA, but that's the only protein I'm going to be able to make. bacteria or prokaryotes, they kind of do things in batches. And so what they have this system where they have operons, where you can turn on a batch of proteins. And so you make one messenger RNA. I want to kind of pause for a second here. Whether or not you can call this messenger RNA is actually a little bit of a debate in like the realm of biology. A lot of times people don't want to call it messenger RNA if you don't have the 5' cap and poly A tail and the splicing stuff going on there. But we'll call it the bacterial equivalent of messenger RNA. A lot of biologists will call it messenger RNA. Some of them won't. And so you might have to be ready for kind of either of those on test day. But once you have this messenger RNA from a bacteria, it's a lot longer and it's got multiple genes coded for on it. And so this one gene, this one messenger RNA will have the code for like 5 or 6 different proteins. And so we'll be able to make lots of different proteins from one messenger RNA. So that's what we call polycistronic. Now we are monocistronic. Our messenger RNA codes for just one type of protein. A bacteria, one messenger RNA can code for like 10 different types of proteins. Now they tend to be related, where like, for example, all of the proteins needed to break down lactose, right, which is a carbohydrate. The idea being like, I will turn them all on or I'll turn them all off, depending on if there's lactose around and if there's glucose around, because I'd rather burn glucose if I can, if I'm a bacteria. But if there's no glucose and a lot of lactose, then I just want to make all the proteins involved in lactose metabolism. And so this is what we call an inducible operon. So by having lactose around, we turn on all of these proteins. And so we can kind of like turn on like this factory that's making a bunch of these different proteins that are all related and all necessary together. There's also repressible operons. For example, bacteria need to make tryptophan because they need to build proteins out of tryptophan. But if there's tryptophan around, then they don't need those proteins. If they don't have tryptophan, then they need to make tryptophan. And so tryptophan is a repressible system. And so this is what we call a trip operon. There's a lac operon for lactose, which is an inducible one. There's a trip operon for tryptophan, and it's a repressible one. So if we have tryptophan around, it will silence this, and we just don't make that messenger RNA. If we don't have tryptophan around, then this is going to be active. So the presence of tryptophan represses and silences the process of making this messenger RNA. That's different than the lactose one, where if you have lactose around, that turns on the system. So the question of inducible versus repressible is like adding this thing, does it turn on or turn off the system? Adding lactose turns on the lac operon, adding tryptophan turns off the trip operon.
Yeah, this is something where I would encourage y'all, again, and I think this is because I'm a visual person, this was something that I actually struggled a lot with, the different conditions of, okay, if you have low glucose versus high lactose, or high glucose, low lactose, or high glucose and high lactose, which one is going to be, are we going to be transcribing the lac operon or not? And I think this is where even doing a system of arrows can be really helpful. And if you were to get a system like this or an operon on test day, you have a scrap sheet of paper, right? You have your wet erase notebook. I would encourage you to make use of it because it can be really tough to keep track of the directions, and when there are a lot of different possibilities, I think it's important to be comfortable with the kind of conceptual framework and then also how they can apply it. Even if it's, we've talked about this in the past, even for some of the physics material, they may not ask you, there will be some cases where you have to plug and chug. There will be some cases where you can answer the question using your conceptual framework and understanding the general relationships. This is one of those where the latter can be really helpful. And just being able to keep track of directionality, I think, is pretty darn important for this.
Yeah, I do think there's kind of a good question of like why is this so different, right? Like the transcription, like in a way, a lot of times the prokaryote stuff kind of makes sense, right? Like if I can just turn on all the proteins all together, like why don't we do that in eukaryotes? Why this much more complicated system of Like, oh, if I want to make the enzymes to break down lactose, I have to make like multiple different messenger RNAs to make multiple different proteins going through that. Like, why is that a better system? A lot of times we want to imagine eukaryotes as being better, right? We're more advanced than bacteria. Well, it comes down to like we are much more complex, like in terms of cells, right? Bacteria, they have like one cell type, right? Like they don't have tissues, right? They don't have like a digestive system. Like they don't have like, oh, we have all these different types of muscle, like skeletal muscle and cardiac muscle and smooth muscle. But we do. And so in our, like you can imagine, like a lot of muscle is going to need similar proteins, right? But not exactly the same proteins and not exactly the same proportions of those. So like skeletal muscle might have this one protein that's involved for muscle, but it needs like much more of it than like smooth muscle does. And so by having the monocystronic system, it allows us to like more fine tune, like, okay, we need like 80% more of this protein, but only 12% more of this protein and 16% more of this. And like this should actually go down 15%. And by like doing all of that, it allows us to like kind of like fine tune so the different cell types, the different tissues are going to have like different levels of proteins. And so like you might see one protein found in all muscle types, but like how much of that protein is in there is actually a little bit fine-tuned and different. And prokaryotes don't need to worry about that, right? So for prokaryotes, their system is way more efficient, right? It's like, do I need this or not? And I'll turn it on or turn it off. Versus, and like there's actually some benefits to that. It's actually more efficient energetically to do that, rather than building like lots of different messenger RNAs. Like let's build one and just kind of like go through that. And be able to kind of batch things out, right? You can kind of think about it as like, you know, if you order something from a shop and they're doing like, oh, they just batch stuff out. Like there's not a lot of customizations that they're going to be able to do. They're not going to kind of like fine tune stuff. Like imagine that you are ordering something from like Walmart or something like that. Like Walmart isn't going to be like, oh, you want this chair, but with these modifications. Like, no, they're not going to do that, right? They're like buy the chair or not, right? We have too much stuff to do. And they value the efficiency versus, you know, you go to like a custom furniture shop, they'll be like, okay, we can make this happen and kind of adjust and go through that. It's not as efficient energetically, or it's not as efficient in terms of time, but it is more efficient in terms of like getting like a more complex, complicated system. And so as a result, prokaryotes can go a lot faster a lot of times in terms of building proteins and things.
Yeah, part of that is also where there are some additional differences in translation that have to do with their, you know, efficiency. And going back to the example that you used here and in the past, it's the actual process of building the chair. So remembering that the central dogma in biology in general is going from DNA to messenger RNA or some form of RNA to the protein. And so translation is when we go from that, in this case, bacterial equivalent of mRNA, the actual protein. And one of the differences is where do the ribosomes bind and how do they find out where to bind? And so in prokaryotes, specifically, there's the Shine-Delgarno sequence. And this is, there's some part of this that is conserved across bacteria in general. And so it's 6 bases. This is going to sound really weird, but it was the way I learned it. It's ag. And so I think this one will perhaps resonate with you, Phil, with your uag. And the sounds you make for the stop codons. Mm-hmm. But this is a component that's conserved, and there are some smaller differences in between. But this is where the ribosomes say, okay, this is where I need to bind before I get to my start codon. And another one of the differences that goes back to this efficiency is that prokaryotes don't have to wait to start the process of translation. They don't need to get any messenger RNA adjusted. They don't need to do those modifications. They don't need to get them out of the nucleus. And so right away, you can actually start translating into a protein as you are transcribing. And so this allows for faster production of the proteins that not you, but that the prokaryotes will ultimately need. And so it's another way that our eukaryotic system does have more control, but is slower. And that's where that customization versus speed balance that you've been mentioning comes into play.
Yeah, it's super interesting because in the prokaryotes, everything's just happening in the cytoplasm. So like the ribosomes are in the same place as the proteins that are building the messenger RNA. And so the messenger RNA, like you said, for prokaryotes is so much like bigger that like you can kind of like build and like we've got this one gene already that like is on a messenger RNA. We haven't finished making the messenger RNA because it's a lot longer, kind of like going through this, and maybe it's got 15 different genes on it that need to be turned into proteins. But you can have ribosomes go ahead and hop on and already start translating this. So in eukaryotes, obviously, like we have the 5' cap, and that's what our ribosome binds in order to, and then it like goes down until it hits a start codon, and then it starts making protein, and then when it hits the stop codon, it falls off. Like that's not going to work in a prokaryote because if you bind like, first off, there's no 5' cap, but if you did bind like the 5' end of this, and then you went down to the start codon, which is still AUG in bacteria, and then you started to build the protein, and then you went to the stop codon, and then you fell off, you're never going to get to the other proteins. Like this messenger RNA has tons of proteins on here. And so what you need is something for the ribosome to bind to that can be like in the middle of this RNA strand, in the middle of this messenger RNA. And so we have our 5' cap because our messenger RNA only has one protein, so just bind the 5' in and you're fine. Just go until you hit the protein and then you're done. But the bacteria RNA, messenger RNA, has to have like a sequence before each protein that we need to make. And that is our Shine-Delgarno sequence. So as a result, the ribosome also has to be a little bit different, right? Because the ribosome in us bind 5' cap. The ribosome in prokaryotes binds the Shine-Delgarno sequence. And so their ribosomes are a little bit different than ours. We have the 40, 60, and 60 subunits that combine to form the 80S subunit for the ribosome, which has got the small and large subunit, small being the 40 and the large being the 60, combines to be 80. And the ribosome in the prokaryotes, their stuff is a little bit smaller, so they have a 30S is their small subunit and 50S is their big subunit compared to the 40 and 60 for us. And they combine to be the 70S subunit. I always just remember that bacteria are weird. They're odd. So they're the odd numbers, the 3, 5, and 7. So the 30, 50, 70. And we are normal. We are not odd. And so it's 40, 60, 80 or 4, 6, 8 for us. That's a good way to kind of keep that straight.
That was also the way I learned it, just with bacteria being odd and then us being even and normal. We are evened out by all of our different tissues, and we're a little bit more balanced. So,
no, I was just going to say there is one thing. I know I said that they both start at the same start codon, AUG, but for us, that codes for methionine, and for bacteria, they have a weird modified methionine. It's an informal methionine. That's N as in nitrogen and formal as in FORMYL, like a formal group, like formaldehyde or formic acid. So it's an informal methionine. Whenever you say that, I always just imagine like a methionine wearing like sweatpants. It's the informal methionine. But yeah, so they also have a slight difference there with their start codon. We both use methionine, but they have a modified methionine.
So we've chatted about the differences in terms of the general cell structure. We've talked about the idea of plasmids, and we've talked about differences in transcription and translation. But there are other things that bacteria can do that doesn't happen between eukaryotic cells. And this is the idea of lateral gene transfer, which is essentially one prokaryotic cell can transfer, or in one way or another, the genetic material is transferred from one bacterial or prokaryotic cell to another. And this is really important when we think about, just from a future medical perspective, and you guys as future physicians, this will become incredibly important, even if you're not diving into the actual, which version of lateral gene transfer is happening. But this is the basis for a lot of bacterial antibiotic resistance, because if you have bacteria that are resistant to one antibiotic and their genetic material can be transferred to another bacteria, it's a recipe for some difficulty. And so there are 3 different ways that prokaryotes and bacteria specifically can transfer their genetic material from one to the other. And the first one is transformation. And so this is a situation where the cell starts to degrade. And I think of it as the transformation. The F stands for final, because it is the end of the life cycle. And so this cell can lyse, essentially explode, and its genetic material is now present in the environment where other bacteria can essentially pick it up. And so this is, I think of it as just that final transformation and that picking up of genetic material. And so that is one way that bacteria can develop antibiotic resistance.
Yeah. And I like that you're hammering this antibiotic resistance, because this lateral gene transfer is super weird if you are used to like eukaryote stuff, right? We only do vertical gene transfer, which means like if I have genes, I can pass them to my kids. That's pretty much it, right? I can't pass genes. If somebody has a like cystic fibrosis and they have a messed up chloride receptor protein, like something's wrong with their gene, I can't just give them a copy of my gene and like now they're fine, right? Like that would be a lateral gene transfer where it's being passed between individuals that aren't like descendant of each other. And so this is huge in bacteria, where you might end up, one bacteria might have resistance to some kind of antibiotic, and that gene can transfer not just to other bacteria in the same species, like from one staphylococcus to another, but it can also move to other types of bacteria. And so now this other, like staphylococcus, the gene for this like resistance has moved between that to streptococcus. So now we have a strep bacteria that is now resistant to that antibiotic. And so this is kind of weird and dangerous and kind of scary from like a biological perspective, where if like one bacteria figures something out, they can spread it to other bacteria, even ones that aren't like related, like in terms of descendants. This transformation one also shows up a ton in research, where you will see like, oh, we transformed blah, blah, blah. And what they mean is like, we're trying to study this certain like protein, for example. And so we put that protein just on like a plasmid, which is the bacteria equivalent of a chromosome. I realize that is also a really big difference. Like the DNA for prokaryotes is a circular strand of DNA called the plasmid, versus we have chromosomes. But you could put like your gene in that and in a plasmid, and then throw that plasmid in a petri dish, and then throw some bacteria in the petri dish, and the bacteria will just pick up those genes. And so that's really useful if you're trying to like study something. For example, like if you're trying to study some protein normally found in Ebola, like I kind of like as a researcher, I kind of don't want to play with Ebola, right? Like if I'm going home to my family, like oh, I don't want to like spend my day messing with Ebola. But I could look at one protein, put it in a plasmid, and then put that in a petri dish with another bacteria. And now that bacteria has this Ebola protein. And so I can study that protein in a safer way. So this tends to happen a lot.
Yeah, something else that's really cool, and that if they gave you a passage about this, you're probably thinking, well, how do I know which bacteria actually picked it up? And so something that they can do is they can have the gene of interest and then an antibiotic resistance gene together. And then what you can think about is when the bacteria picks this up, you can actually not screen out, but basically remove all of the ones that didn't pick this up, because you add the antibiotic that the ones that picked it up are resistant to. And so this is a way where you can actually almost clean up what you're interested in looking for. And so there are a lot of different applications for this. And if you were to see something like this on test day in a passage, you want to be able to understand how it works and why it works.
Yeah. This is one of those things that they will test just like directly, but also understanding it helps you read the passages. And that just makes it a lot easier to kind of deal with the passages. So I always feel like the best is always examples. Like, so let's say I wanted to study, there's a passage that I've came across fairly recently about this Ebola glycoprotein, which is what I was kind of talking about, Ebola. But you could take that protein, put it in a plasmid, and then also put like an antibiotic resistance in that same plasmid, throw that into a petri dish with this other bacteria, like say like some stomach flu, or like not flu, because that would be a virus, but like some other bacteria. There's also Ebola is a virus, now that I'm thinking about it. So that's why I'm thinking this one. Okay, forget this. So Ebola would be a virus, but like some other bacteria, you take like some protein, antibiotic resistance, throw it into a petri dish, throw the other bacteria in there, and then you throw the antibiotic in there. So only the bacteria that picked up that plasmid will survive. So the only bacteria in that plate are gonna have this protein that you're looking at. And so you can kill off anything that didn't pick up that by using that antibiotic. But like a part of this, like for like the gene transfer, if it's like, wait a minute. So like for transformation, if it's only cells that have died and like lysed, like that's the only way for them to do this lateral gene transfer. Like that's kind of not as big of a deal, especially for like antibiotic resistance, right? Because these will only transfer between things that are being killed. And so like if an antibiotic kills something, then those genes go to other bacteria. Then it's like, well, okay, well, the antibiotic killed this thing. So if those genes go into other stuff, that's fine because that obviously didn't protect from that antibiotic. But it's possible for bacteria to transfer their genes without dying. And so another way they can do this is conjugation, where a bacteria will like make a copy of its plasmid and then inject that plasmid into another bacteria through like physical contact. This conjugation, there's an F factor, which can be found on the bacteria. It's like the fertility factor is what they call it. Some of the language with this, the way that this works is like a bacteria develops what's called a sex pilus. It's not really sex, but like basically there's like a long tube that pokes into the other one and then genetic material is transferred through that. And so there's a lot of like vocabulary that's like, this is kind of like sex, right? Where something like injects and then transfers DNA through this. It's not obviously like sexual reproduction or anything like that, but it is this lateral gene transfer. And so I always like to think of like conjugation reminds me of conjugal visits, in case you are curious of what that is. So in prison, if there's like a husband and wife that one of them's in prison, like they will allow them to get together with some privacy, and those would be conjugal visits. And so I always think about that with conjugation. And so that's kind of what's happening here is like a bacteria, they have to be in physical contact with each other though, where one bacteria runs into another bacteria and it can transfer genes. And they have to have the F factor, which is this ability to that allows them to form this sex pilus and do this lateral gene transfer.
Yeah, I don't think we can emphasize this enough in thinking about y'all as future physicians. What we call antibiotic stewardship or using antibiotics wisely is so incredibly important. Like it is there, we have a finite number of antibiotics and we are still learning about new bacteria. We're still watching bacteria develop resistance. You can think about how with conjugation, bacteria that are, resistant to an antibiotic right then and there can get other bacteria resistant to that particular antibiotic. You can think about with transformation, something that's, lysine might have a resistance to a different antibiotic that isn't being used. But now all of a sudden you have bacteria that are resistant to this other antibiotic. And if you go to use that other antibiotic at some point, you're out of luck, right? And you think about Staph aureus. This is something that this is something that I think is just generally useful. Used to be susceptible to an antibiotic called methicillin, but now there's so much resistance to it. And as you guys working in the medical field, a lot of us will end up being colonized by this. And so you think about how for your health, for your patient's health, it becomes incredibly important. And so while you may not see or talk about these exact mechanisms when you're a physician, you need to understand these ideas because it's going to influence how you provide care to your patients. So I don't think we can stress this enough for your future careers.
it is something super interesting going into that. So I know I've talked about this before, but I have grown up in the like farming community. And honestly, farmers use a lot of like ranchers and stuff will use antibiotics. Honestly, like it should be more regulated than what it is. Because they obviously don't want all their cows to die. And so they just use like antibiotics often. And like if there's like anything kind of going on. And I think that sometimes this can cause some issues. But you guys probably as physicians won't be in charge of that. But it is important to like kind of understand how this stuff works, and like use of antibiotics inappropriately is like could lead to people dying in the future. And so exactly that stewardship is very important.
Yeah. So we said at the beginning that there were 3 different forms of lateral gene transfer, thinking about that horizontal gene transfer. And you're probably thinking, well, you mentioned 2. Where's the third one? Mm-hmm. And the third one is going to be transduction. And so for transduction, these bacteria need a little bit of help. And so instead of them picking up the plasmid from their surroundings, instead of one bacteria transferring it through conjugation to another, basically a virus gets involved. And so we call that a bacteriophage. And you can think about situations where a virus can actually insert, but also pick up not bacteria, can pick up genetic material, and it can go and then infect another bacteria. And so now all of a sudden you've transferred this genetic material, this perhaps antibiotic resistant or other genetic material, a gene that codes for something else, from one bacteria to another. And this is something that's incredibly important because it's oftentimes, just thinking about our future career, someone can have multiple infections at once, right? And you won't always know it. And so this is another way that we can see this gene transfer that can be important and that probably has implications that we don't even realize. And there might be situations that are happening that we just haven't discovered yet. And that's almost the beauty and the madness of medicine and science in general. It's we don't know what we don't know. And as we continue learning, we can, do our best, keep up. But having this mindset of we need to learn more can be incredibly beneficial. And so if there are any folks that are thinking of doing MD, PhD and thinking about your research, and you're going to love it, Phil, then, just thinking about what kinds of studies are interesting to you, what kind of topics are interesting to you. And don't be afraid to also think what could be actually useful or applicable, clinical versus basic research. All of that is incredibly important. And if folks are thinking about public health, there are a lot of implications with what we've just talked about with public health and kind of the use of antibiotics as well. So there are a lot of different places where medicine and other career components can come into play. And so if any of you guys are interested in those things, I would encourage you to explore, because there's just so much out there that we can do as future physicians, and that you guys can do as future physicians, that I want to encourage you to explore.
Yeah. This, like, the transduction thing for me is the most interesting of all of these. Like, it's super, because it's this weird kind of like it requires viruses, like I shouldn't use in conjugation, because that's going to make people think conjugation. It requires viruses to work together with bacteria, and working together is probably not the right term here. Like, because what viruses will infect the bacteria, like hijack the bacteria's machinery, make tons of copies of the virus proteins, including like the capsid that's like injecting the DNA. And then they'll make copies of the virus DNA and they'll put those together. And so like we'll release this new virus, which can like bind another bacteria and inject the virus DNA into that bacteria. But when the virus is forming that packet with the virus DNA, if that's occurring inside the bacteria, it might mess up and put the bacteria's DNA inside that virus. Because everything's kind of floating around in the cytoplasm. So there's some like viral DNA here, but there's also like a chunk of bacteria DNA. And so when the virus is forming, it'll form around that. And then when that virus goes and injects another bacteria, it's not injecting virus DNA, it's injecting the original bacteria's DNA into that. And that's super just weird, right? This like having a virus act as a vector for transmission between like genes between different types of bacteria. This is also something that like this should be kind of like tinkling in your brain of like, this seems like humans can get viruses. Like, is there something that like, is there some way we could do that? And actually a lot of research and a lot of stuff is built around this as well. Definitely all of the first gene therapy stuff that we've done. Note that like viruses will inject their DNA into your cells, right? And so if for some reason what's inside that virus might not be virus DNA, it might be something else, like that DNA might be able to get into you, especially the lysogenic viruses, which can incorporate like DNA into your DNA. We did an episode on that a while ago. It's my favorite topic, or it's my favorite title from any of our episodes. The episode title is, if you are listening to this, you've probably got herpes, which is factually correct. Like, you probably have herpes. But listen to that if you want to learn more about lysogenic viruses. But you could think that like, okay, this could lead to some like a virus carrying some genes injecting into yours. This may be like that person who is missing like who had the mutated chloride receptor and therefore has like cystic fibrosis. If we could somehow engineer a virus to inject the correct gene into that organ, into that person, it could cure them. Now we'd have to inject that into all of their lung cells, not just one cell, but like all of them, which would be kind of hard. And that can also lead to other problems, because what if we inject it into the wrong spot in the DNA and we inject it like in the middle of like P53, which is a tumor suppressor gene, and we knock that out? Like, that's going to be a problem, could lead to cancer. So this doesn't work super well, but it has been seen that genes have been transferred between humans, or from a bacteria, that DNA has gotten into a virus and been injected into human DNA. This is actually one of the biggest piece of evidence for evolution. If you are looking into like evolutionary theory, there are certain bacteria genes that are in your DNA. They don't do anything in you because you're not a bacteria. But it's kind of interesting that you will see that same bacteria gene in chimpanzees. And so the chimpanzees and us will both have the same bacteria gene injected in the same spot in our DNA, which implies that we have some common ancestor that like they had bacteria DNA injected into that ancestor's DNA, and then it kind of split off after that. But you don't see that in the other apes. So that's another piece of evidence. Like, oh, we're a little bit more closely related to chimpanzees than we are the other great apes. So I don't want to kind of spin off into evolution, but this whole idea of transduction and this lateral gene transfer is the foundation of a lot of gene therapy stuff. And so a lot of future medicine is kind of built off of that, especially with things like CRISPR, which allow us to be much more precise with where we're cutting DNA. And it is theoretically possible to like start to be able to fix people's genetics, like if they have like a genetic mutation that's causing a disease, by doing some kind of lateral gene transfer. And the only one that we really see occur in humans is transduction. But even then, it's really, really rare to see that in humans. So I don't want you to think like, oh, humans do a bunch of lateral gene transfers. Like, no, we don't. Like, this is something that like we had an ancestor who it happened to once, and it's kind of interesting. But it is theoretically possible.
Yeah, these topics are ripe for discussion, for research, and for MCAT questions, which is why we're doing this podcast episode. Just recommend that if you're a visual person, take a look at some images and some comparisons, make your own. I'm a very big proponent of review sheets and study guides that you make yourself. And if this is something that you have the time for and you're interested in, feel free to do some additional research. As a reminder, the podcast, we go into some detail, but it is not exhaustive. And I think it's important to recognize that every so often, because you do still need to do some additional reading, do some additional prep for all of these topics. But our goal is ultimately to help you out and to get you started on your MCAT studying road and help you through it.
Yeah. To just kind of wrap up, just to reiterate, focus on the differences, right? What are the differences? There's a reason we hammered like gyrase and Shine-Dalgarno sequence and the 30, 50, 70 ribosome, because if we make anything that messes with those things, like if I make a drug that messes with informal methionine, it's going to kill bacteria, not kill humans. That sounds like a useful antibiotic. So definitely be thinking about that as you're going through. Think about how the AMC is going to test this, like in the context of some antibiotic or in the context of some bacteria developing resistance to an antibiotic. Because this is what a lot of the backbone of medicine is built upon. And so it's really important to make sure that you understand this well, not just for the MCAT, honestly, but also to just be a great physician.